Hello everyone. Today in the subject of medical microbiology, we are going to study about bacterial pathogenesis. This particular topic, the main objective is to give you a brief introduction about bacterial pathogenesis, what are the dangerous things that are involved with it, and what is the working mechanism of this particular topic. So outcome will be you'll be able to understand what are the intricate details that are employed in the different working mechanism of pathogenesis and what are its essential points. One must realize that pathogenesis of bacterial infection, like pathogenesis, is anything that harms you. Okay, whatever the thing is there, or any sort of infection that is harmful for you. We would focus on how bacteria cause disease to humans, to human beings. This process of causing disease is termed as pathogenesis. So you must also understand the term disease has been derived from ancient Greek and Latin word dis-ease, means discomfort in your ease, means disease, and pathogenesis is causing of disease, the mechanism or the procedure of causing a disease, anything or any microorganism that is harmful in nature is simply called as pathogen. Pathogenesis is a multifunctional process which depends on the immune status of the host. So it is a multifunctional process, okay, and multi factorial process which depends on the immune status of the host. Like better the immune system, stronger the immune system, less vulnerable that particular host is towards the pathogens. Okay, in simpler language, we can elaborate it as the stronger the host, the more stronger the immune system. If the host or the individual is immunocompromised or his or her immune system is not functioning up to the mark, then that individual is much more prone to the attacks from the pathogens. The nature of species or strain virulent factors and the number of organisms in the initial exposure. So, virulent factors or the virulence uh, influences the ability of a pathogen to cause some sort of infection in a host. So, it involves different mechanisms like releasing toxins, releasing enzymes. Okay, so each species has its own virulence factors and its own mechanism of infection and its own target tissues. Okay, not every bacteria targets everything. Every bacteria has a certain type of um, working mechanism. Because of those things, they target the that very particular tissue. So, the number of organisms in the initial exposure. A limited number of bacterial species are responsible for the majority of infectious disease in healthy individuals. Right? A number of bacterial species are responsible for a majority of infectious diseases in healthy individuals. If we see the spectrum of bacteria or bacterial population, we have two extremes. One side is extremely useful and beneficial for the human and non-lethal, and the other side is extremely pathogenic in nature, right? So what they're trying to tell you here is that not all bacteria they are dangerous in nature, right? And not all of them create a lot of trouble for human beings. So due to a large number of bacterial species are responsible for majority of disease. So Those small minority of bacteria that are pathogenic in nature, they cause a lot of trouble to the human beings or the healthy individuals. Due to the success of vaccination and antibiotics and effective public measures, until recently, epidemics were felt to be a thing of past. Earlier, epidemics were used to be due to a lot of pathogens that were easily curable or easily treatable diseases. But lack of hygiene, uh, lack of sanitation facilities used to give rise to many other diseases substantially that can be easily eradicated by the usage of simple measures. But as the technology is increasing, everything is getting crucial day by day. Antibiotics have also been a great achievement. So all these things have been able to eradicate a lot of diseases from the planet of face of the planet Earth, and it has been really a thing of the past nowadays. Due to the development of antibiotic resistant organisms, this situation is changing rapidly. Now, the problem with the antibiotic medication is that you have to complete the course. Okay. And what people generally do, okay, they took one dose and they felt better and they left the medicine. So, whatever type of medicine you are taking, whatever things you are doing, right, and from whatever disease you are suffering, you always have to look into the context that you always have to complete the course of the drug. Okay. If you were supposed to take it for 3 days, then take it for 3 days. Not like okay, I just felt better in one day and you left it. So this scenario leads to the uh, antibiotic resistant organism. They are also known as or pathogens that are offering drug resistance. So earlier these pathogens were not able to withstand against the antibiotic drug, but nowadays because of these things they were able to not only withstand the drug and they were also able to face the drug crucially. So there is a need for the development of new drugs because these recent drugs are now getting or facing a lot of resistance from these pathogens nowadays. All humans are infected with bacteria, the normal flora. Actually, they are talking about the microbiota that is normally found in the human body, living on their external surfaces, including skin, gut, and lungs. Because our body is fully covered with bacteria, right? Whether it's our skin, gut, lungs, ears, or whatever you say, wherever you see. But these bacteria, they are known as microbiota, and they do not offer any difficult situations because they occupy the space in our body and they get advantages from our body. So whenever external pathogen attacks our body, these pathogens, first of all, they do not offer them space to make themselves comfortable and stable. And after that, they have to face the competition with respect to the microbiota of our body. We are constantly also exposed to bacteria, including air, water, soil, and food. Normally, due to our host defense, most of these bacteria are harmless. Okay, we are constantly being bombarded by these bacteria. Okay, whether we are exposing ourselves to air, water, to soil, or food, whatever is the case, our body is constantly combating of these pathogens or these bacteria. And because thanks to our defense system, most of these bacteria are repelled off, but majority of the bacteria are also harmless in nature. In a compromised patient, and they are talking about immunocompromised patient, Those immune system is not up to the mark because of weakness or any sort of illness, whose defenses are weakened. uh, these bacteria often cause uh, opportunistic infectious disease. Okay, so these bacteria, they are always in they are always seeking some sort of opportunity, right? And when this individual or the patient will become weak, we will immediately attack it. So, opportunistic infectious diseases, when entering the bloodstream after surgery, catheterization, and other treatment modalities. So, whenever you see such treatment, what you generally see, you see that whenever a person is going through some sort of surgery, okay, and some sort of disease, when the immune system is really weak, so these opportunistic infectious diseases, when entering the bloodstream after surgery, catheterization, and other treatment modalities, so they always try to infect the individual, and they cause a lot of trouble for the individual who is in an immune compromised situation. When initiated in the hospital, these infectious disease are referred to as nosocomial infections. They are in other way you can say that they are also known as hospital borne diseases or hospital borne infections, right? So one should always try to make it try to avoid visiting hospitals frequently and clinics frequently, because most of the time all the diseased or sick individuals, they gather in these places. Okay, and although these places are very clean and secure, I agree with that. But you are more likely to catch some sort of secondary infection from some individual who was suffering from a contagious disease. So, in the long term, one must be extremely careful and cautious if he or she visits hospital, and one must always be careful when you are dealing with any of such things. Some common bacteria found in the normal flora include Staphylococcus aureus and epidermidis, and bacterioids and Enterobacteriaceae found in the intestine, the latter in much smaller number. So here you can see the different phases of the pathogen and how it operates. So here they are showing the two extremes. Some bacteria are more contagious during incubation period. Some bacteria are more infectious during invasive period. So they are mentioning two things here. Invasive period, when the bacteria invades your body cells or your body. So some bacteria are extremely contagious or extremely disease causing during that period. And once the bacteria has invaded inside your body, uh, the incubation period starts, where it generally increases its population or number. So some bacteria are more contagious during the incubation period, right? So there is a big difference between the two. So what does this scale represent? Incubation period length varies. Okay, some bacteria have incubation of 15 days. Some bacteria have incubation period of 10 days. Onset of illness and prodromal phase. So, at what stage they have started to release of their toxins? There might be exotoxins, there might be endotoxins. On what period they are releasing and in what amount they are causing irritation in the body. Period of invasion, length varies. So, some pathogenic bacteria have short duration of time. Some bacteria have very long duration of time. Uh, Convalescent period, recovery phase. And after causing such infection, these bacteria they particularly leave the body and or leave the body cells on body tissues and try to go somewhere else and infect someone other. So this is the whole scenario that is followed when infecting some individual. So coming on to the pathogenicity. Pathogenicity is the capacity to initiate disease, right? So what is the main capacity to start a disease? What is the main potential that bacteria needs to have in order to switch on the onset of a disease? It requires the attributes of transmissibility or communicability, right? So it should be able to be transmissible in nature, and it should be able very much able to be communicable in nature, right? So it can be transferred from one individual to another, and it should also be highly contagious in nature. From it can be easily transferred from one host or reservoir to a fresh host. Survival in the new host, infectivity, or the ability to breach the new host defenses and virulence. So, what they are talking about here, reservoir reservoir to a fresh host, transferability, survival in a new host. So, once the bacteria enters the body, it has to survive inside it, and infectivity. So, how infectious it is proving itself to be. Its ability to breach the new host defenses and virulence. So, how that very particular pathogen is going to evade the host defenses and uh, use its various virulent factors. Ability that is multifunctional and denotes the capacity of a pathogen to harm the host. Right. So, what are its multidimensional qualities due to which it is going to harm a host in the long term? So, virulence in the clinical sense is a manifestation of a complex bacterial host relationship. Okay, so it is a complex relationship between the pathogen and the host, in which the capacity of the organism to cause disease is considered in relation to the resistance of the host. Right. So, virulence factors mainly involve the capacity of the host to cause some sort of infection. But it also comes in hand with the capacity or with the ability of a host to defend itself, to fend off the attacks. Okay, so it is considered to be a relation between the bacteria that is pathogenic in nature and its host. So, a type of bacterial pathogens. Bacterial pathogens can be classified into two broad groups: primary and opportunistic pathogen. Right. So there are two types of pathogens. One is primary pathogen, and other is opportunistic pathogen. So primary pathogen are capable of establishing infection and causing disease in previously healthy individual with intact immunological defenses. So they are able to cause some sort of infectious disease in the individuals who were previously healthy and their immunological defenses were intact. Okay. However, these bacteria may more readily cause diseases in individuals with impaired defenses. So they can also readily infect the individuals who are not at all that much strong, or they do not have a much better immune system radically. So, opportunistic pathogen rarely cause disease in individuals with intact immunological and anatomical defenses. So what they are trying to explain here is if the individual is in good health, if he or she is readily uh, in a healthy condition, in a healthy state, his or her immune system is up to the mark, and there are no anatomical uh, differences or weaknesses based on their immunological reasoning also, then there's no problem, then there is no issue regarding to the uh, health and other consequences. So, uh, opportunistic infection, they do not affect the individuals who are physically strong and their immune systems are up to the mark. Only when such defense are impaired or compromised as a result of congenital or acquired disease or by the use of immunosuppressive therapy or surgical techniques are these bacteria uh, able to cause disease. So, when such a strong individual is there whose immune system is not at all weak, but uh, s- due to some reason, somehow, the immune system is impaired or immunocompromised situation arises, then these opportunistic pathogens they seek a chance. Okay, uh, and this chance because of might be because of some sort of immunosuppressive therapy or any sort of surgery or any sort of condition that can be possible as a result of some case. So these opportunistic pathogens they always seek such a chance. Many opportunistic pathogens, for example, coagulase, uh, negative Staphylococcus, Escherichia coli, are the part of the normal human flora. So most of the bacteria they are also part of the normal human flora, or also known as microbiota, right? Are and are carried on the skin or mucosal surfaces where they cause no harm and may actually have beneficial effects. So it is a mutual benefit. You offer them shelter and they offer you some sort of vitamin synthesis, right? So this is a mutual relationship between the bacteria and the host by preventing colonization by other potential pathogens. So, what is the main advantage of having microbiota in your body is that they offer no space to the invading pathogens. So, it is very difficult for them to establish themselves and to anchor themselves in our body. So, it is extremely rare and extremely tough for a pathogen to enter inside our body and to establish itself. However, introduction of these organisms into anatomical sites in which they are not normally found, or removal of competing bacteria by the use of broad spectrum antibiotics may allow their localized multiplication and subsequent development of disease. So, uh, introduction of these pathogens to the anatomical sites which they are not normally found. So how does this take place? Because when some sort of surgery is going on, when any of such procedures are going on, the removal of competing bacteria, removal that leads towards the removal of microbiota that is normally found on the skin by the use of broad spectrum antibiotics. Antibiotics does not differentiate between the microbiota of the skin and the pathogen. So it allows the localized multiplication of and subsequent development of these diseases. So these pathogenic microorganisms they do get a chance in the long run to establish themselves. The above classification is applicable to the vast majority of pathogens. However, there are exceptions uh, and variations within both categories of the bacterial pathogen. Right. So, different strains of any individual bacteria species can vary in their genetic makeup and virulence capacity. So, each species of bacteria uh, has its own speciality, has its own property, right? For example, the majority of Neisseria meningitis strains are harmless uh, common cells and considered opportunistic bacteria. However, some hypervirulent clones of organisms can cause diseases in a previous healthy individual. So, some bacteria are not at all harmful to you, but due to some mutation, due to some changes, they become hypervirulent in nature. Okay, then these bacteria can really be harmful. Conversely, people vary in their genetic makeup and susceptibility to invading bacteria. Some people are much more prone to catching on some sort of bacterial infection, while others are not. So, the state of immune system, it varies greatly among the individuals. Some individuals, they have a very good immune system. Some individuals do not have that much a strong immune system. Okay, so it varies from person to person. For example, Mycobacterium tuberculosis is a primary pathogen but does not cause disease in every host it invades. So, Mycobacterium tuberculosis is a opportunistic pathogen. Okay, it will invade your body, but if your immune system is up to the mark and your body is really strong, then it won't cause any disease in you. But if the host becomes weak and immune system becomes compromised, it will immediately attack the host. And until unless that stage arrives, it will be there inside the body in a dormant phase. Now, coming on to the Koch postulate. Koch forward four criteria designed to establish a causal relationship between a causative microbe and a disease. Right. So, Koch was a scientist who was trying to uh, find out some causal relationship between the pathogen and a disease. Okay. So, the postulates were formulated by Robert Koch and Frederick Loeffler in 1884 and redefined the and published by Koch in 1890. Koch applied the postulates to establish the etiology of anthrax and tuberculosis, and now have been generalized to other diseases also. So, this work laid the foundation, and that has significantly made us able to formulate the vaccine for coronavirus also. So, he gave the basic principles that are applicable to this day, still functional in nature. Okay, so first, the organism must always be found in humans with infectious disease, but not found in healthy ones. So, the pathogen that is causing disease is correctly identifiable in the individuals who are sick, but it is completely absent in the individuals who are healthy and strong. The organisms must be isolated from humans with infectious disease and grown in pure culture. So these organisms should be isolated from humans who are suffering from disease, and these pathogens have been identified as uh, the contagious pathogenic organism, and they should be grown separately in a pure culture. Third, organisms isolated in pure culture must initiate disease when re-inoculated into susceptible animals. So, when these organisms are being re-inoculated in order to initiate the disease, okay, the pure culture is re-inoculated into a susceptible host. It should completely be able to cause or to introduce the disease. Fourth, the organism should be re-isolated from the experimentally infected animals. So the organism that has been re-inoculated should be completely separated from other experimental animals, experimentally infected animals. So postulates 3 and 4 are extremely important in definite proof of the role of agent in human disease. However, this depends on the ability to develop animal models that resembles the human disease. In many cases, such models do not exist. So here are the cost postulates diagram. The first thing you can see here, the suspected causative agent must be absent from all healthy organisms but present in diseased animals. So here you can see healthy organism and its blood is being examined under the microscope, and then it is being cultured in the petri plate. Then diseased organism that is not at all active, and when its blood is inspected under the microscope and cultured in petri plate, you can see the causative agent present. So what is happening in the second phase? Uh, the causative agent must be isolated from the diseased organism and grown in a pure culture. So what is the next phase? Here you can see the injection with cultured agent, and as a result of that, the individual is falling sick. The cultured agent must cause the same disease when inoculated into a healthy susceptible organism, right? So when you inject the healthy uh, animal with that, it will cause disease. Fourth, so causative agent has been proven to be present in the petri plate or your original culture. The same causative agent must be then re-isolated from the inoculated diseased organism. So these are some cost postulates. Okay. So pathogenesis, the process of pathogenesis involves steps beginning with transmission of infectious agent or infectious pathogenic agents. Here we are talking in the context of bacteria to the host, followed by colonization of the site. So what we have to look into the context is that we have to transmission of that very pathogen and coming into the contact of host and followed by the colonization of that very particular site. After the colonization of host, the bacteria remain adherent at the site of colonization, then invades the host system. So when the colonization of the uh, inside the host takes place, the bacteria remain adherent to the site of colonization, then invades the host system. This step is extremely essential because in the whole following the whole scenario, the bacteria is trying to uh, increase its population in order to secrete some sort of virulent factors, but also it is extremely important because it is very difficult to make itself establish inside the host system because constantly it has to evade the immune system also. So after surviving the host immune system, it is ready to cause disease. So luckily that one bacteria survived the immune system attacks, and as a result of that, it forms many colonies, and it forms many different sites of infection where the number of the bacteria is increasing, and eventually it is causing a disease. So, what are the steps involved in the pathogenesis of bacteria? If we talk about that, what are the initial steps? Then transmission, colonization, addition, invasion, survival in host, and tissue injury are the common steps. So here you can see in the diagram we are talking about some common site that suffer from bacterial infection like eye infection of Staphylococcus, Neisseria, Chlamydia, sinusitis by Streptococcus, Haemophilus, and upper respiratory tract infection because of Streptococcus and Haemophilus, gastritis, Heliobacter pylori causes a lot of trouble, gastric trouble, and food poisoning is as a result of eating contaminated food that is caused by uh, Campylobacter, Salmonella, Shigella, Clostridium, Staphylococcus, E. coli. Urinary tract infections caused by E. coli, other Enterobacteriaceae, Staphylococcus, Pseudomonas, and STDs, the sexually transmitted disease by chlamydia, Neisseria, treponema, ureaplasma, hemophilus, and many skin infections are a result of Staphylococcus, Streptococcus, and Pseudomonas. Pneumonia, community acquired, okay, mainly caused by Streptococcus, Haemophilus, Streptococcus aureus. Atypical, Mycoplasma, Chlamydia, uh, Legionella, Mycobacterium, tuberculosis. Otitis media, Streptococcus pneumonia, right? Bacterial meningitis caused by Streptococcus, Neisseria, Haemophilus, Streptococcus, and Listeria. So these are some common bacterial infection sites that are uh, mostly seen in humans. On these different locations, infections are caused by the pathogenic bacteria. Transmission. Potential pathogen may enter the body by routes various routes, including the respiratory, gastrointestinal, urinary, or genital tracts. Alternatively, they may directly enter tissues through insect bites or by accidental or surgical trauma to the skin. So wherever they are going to see an opening, they are going to proceed to infect your body because they are mostly opportunistic in nature. So many opportunistic pathogens are carried as part of the normal human flora, and this acts as a ready source of infection uh, in, in the compromised host. In case of AIDS, when the skin barrier is breached. So, what happens when the individual is suffering from AIDS? AIDS does not kill the individual, but as a result of this, it readily weakens the immune system. That body is not at all able to fend off the attacks from these opportunistic pathogens, and individual is able to not able to fend off itself, and individual is not able to save itself or defend itself against these attacks. For many primary pathogens, however, transmission to a new host and establishment of infection are more complex process. So here you can see the transmission can occur by nasal, subcutaneous injection, insect bite, stools, urine, and wounds on the of the skin. Colonization. The establishment of a stable population of bacteria on the host skin or mucous membrane is called colonization. So when the bacteria enters inside and it it anchors itself. So the main important thing is to increase its number so that it can readily be able to form a biofilm and protect itself from the constant attacks that it is facing from the immune system. For many pathogenic bacteria, the initial interaction with host tissues occurs at a mucosal surface, and colonization normally requires addition to the mucosal cell surface. This allows the establishment of a focus of infection that may remain localized uh, or may subsequently spread to other tissues. So, uh, bacteria they require colonization to uh, stick to a particular site, and when they are able to stick to that very particular site, then they are also able to proliferate and spread to the other portion of the body. So here you can see the diagrammatic representation of the same procedure. Here you can see the pathogens are coming inside and following their mortality movement, and they encourage themselves on the uh, epithelial cell layer. And with the result of that, after a certain sustained period of time, they go through proliferation and leads towards matrix formation. Okay, and when the maturation takes place, it forms a biofilm, and very much of interest. This bacterial population is increasing its size and number, and after a certain sustained period of time, it detaches the pathogens further in order to infect or in order to cause some sort of infection to the other sites also inside the body. So this was the main thing I was trying to explain to. Addition. Addition or means to stick. Addition is necessary to avoid innate host defense mechanism such as uh, peristalsis in the gut and the flushing action of mucus, saliva, and urine, which removes non-adherent bacteria. Okay, so peristalsis and reverse peristalsis are the movement that readily removes the bacteria, and mucus, saliva, urine also flush out the bacteria from our body. For bacteria, adhesion is a necessary preliminary to colonization, then penetration through the tissues. So, first of all, colonization is also important, but the initial phase focuses on uh, sticking to a site. So, addition is extremely important. Successful colonization also requires that bacteria are able to acquire essential nutrients, in particular iron, for growth. At the molecular level, addition involves surface interaction between specific receptors on the mammalian cell membrane that usually includes carbohydrates and ligands, usually proteins. But on the bacterial surface, the presence or absence of specific receptors on mammalian cells contribute significantly to the tissue specificity of infection. So in this diagram, you can see the material surface, physical, chemical, and mechanical properties. Here you can see the bacteria. It is in stage one, and it is moving around freely. Stage two, it colonizes itself. The proliferated colony, adhesive ligand receptor complexes that are made up of protein, and it establishes itself due to the molecular level interaction with the host cell. Then 3D community peptidoglycan envelope forms all around the mature biofilm. So these are the certainly some stages of infection that significantly infect the individuals. Non-specific surface properties of bacterium include surface charge and hydrophobicity. Also contribute to the initial stages of the adhesion process. Several different mechanisms of bacterial adherence have evolved, all utilizing specialized cell surface organelles or macromolecules that help to overcome the natural force of repulsion that exists between the pathogen and its target cell. Many bacteria express pili or fimbriae, which are involved in mediating attachment to mammalian cell surface. Different strains or species of bacteria produce different types of pili, which can be identified on the basis of antigenic composition, morphology, and receptor specificity. Invasion. Invasion is penetration of host cell. Okay, when a bacteria trying to invade inside the host cell and tissues beyond the skin and mucous surfaces, and is mediated by a complex array of molecules, often described as invasions. The bacteria release certain types of chemicals or molecules that assist in the invasion inside a tissue. These can be in the form of bacterial surface or secreted proteins which target host cell molecules, uh, particularly receptors, uh, once attached to a mucosal surface. Some bacteria, for example, uh, diphtheria and Clostridium tetani, exert their pathogenic effects without penetrating the tissues of the host. These produce biologically active molecules such as toxins, which mediate tissue damage at the local or distant sites. For a number of pathogenic bacteria, however, adherence to the mucosal surface represents only the first stage of invasion of tissues. Examples of organisms that are able to invade and survive within the host cell include Mycobacterium salmonella, Shigella, and others. So here you can see uh, in the first step, bacterial adhesion binds to host receptors. The ligation of a receptor uh, induces downstream signaling events that culminate in the invagination of the host membrane and extension of actin filaments around the bacteria. Third, membrane fuses with the the now enclosed bacterium in a vacuole. So collapse of actin restores the host surface. So this is how the bacteria enters inside the host. The initial phase of cellular invasion involves penetration of the mammalian cell membrane, and many intracellular pathogens use normal phagocytic entry mechanism to gain access. Inside the cell, they become surrounded by the host cell derived membrane vesicles. Many intracellular pathogens escape from these vesicles into the cell cytoplasm, where they multiply rapidly before spreading to adjacent cells and repeating the process of invasion. The ability of specific receptors on host cell defines the type of host cell that are involved. As a result, some pathogens can invade a wide range of cell types, while others have a much more restrictive invasive potential. So the receptors for some of the invasive pathogens have been identified. Virulence determinants. So what are the things that initially determine the virulence capacity? Both primary and opportunistic pathogens possess virulence determinants or aggressions that facilitates the pathogenesis. Possession of a single virulence determinant is rarely sufficient to allow the initiation of infection and production of pathology. Many bacteria possess several virulence determinants, all of which play some part at various stages of the disease process. In addition, not all strains of a particular bacteria species are equally pathogenic. For example, although six separate serotypes of encapsulated Haemophilus influenzae are recognized, serious infection is almost exclusively associated with isolates of serotype B. Moreover, even within serotype B isolates, 80 of serious infections are caused out of more than 100 clonal types. So here in this diagram, you can see the difference, uh, different bacteria in virulence factor. Here you can see exo, exotoxin, right? Virulence plasmids, toxin producing phage, adhesion, adhesion pili, capsule, adhesins, virulent enzymes, lipopolysaccharide membrane, flagella, iron binding protein, virulent surface protein. So these are all virulent factors that are essentially needed for this purpose. Different strains of a pathogenic species may cause distinct type of infection, each associated with possession of a particular complement of virulence determinants. Different strains of E. coli, for example, cause several distinct gastrointestinal diseases, urinary tract infections, and septicemia, meningitis, and a range of other minor infections. Many pathogens produce an impressive armory of virulence determinants. However, their expression is coordinated or regulated by several nutritional and environmental factors. Among virulence regulators, the ability of nutrition, for example, iron, oxygen, suitable temperature, or other growth requirements. Importantly, difference in virulence between similar organisms may be due to additional uh, cryptic, phenotypic, or genotypic variations. For example, some virulence factors are only expressed when in direct contact with host cells. Virulence genes can move between bacteria via special genetic vehicles, for example, plasmids, bacteriophage, and transpons. The horizontally transferred virulence factors, for example, toxins, may or may not transform the recipient bacteria into uh, better adapted or more virulent pathogen. So, survival in the host. Many bacterial pathogens are able to resist the cytotoxic actions of plasma and other body fluids involving antibody and complement in a classical way, or complement alone or in an alternate pathway or lysozyme. Killing of extracellular pathogens largely occurs within phagocytes after uh, opsonization by antibody and or complement, and phagocytosis, circumvention of phagocytosis by extracellular pathogens, is thus a major survival mechanism. Capsules, many pathogens, proteins, and M proteins functions in this regard. Protein A is the surface constituent of S aureus, as well as a secreted product and binds to the Fc portion of immunoglobulins. Bacteria on binding antibody activates the classical complement cascade, which results in the attachment of fragment C3. Phagocytosis occurs after binding of opsonized bacteria to receptor uh, for the Fc portion of IgG or C3 regions. So here you can see bacteria and the host cell. First is invasion, zipper mechanism, contact by the surface of protein, phagocytic cup formation, closure and retraction, trigger mechanism, injects its effectors by TSS, cytoskeleton rearrangement, and engulfment bacteria by a vacuole. So now comes to the survival. Membrane-bound lifestyle prevent lysosome fusion, microenvironment change, cytosolic lifestyle disrupt vacuole membranes by secretion, enzymes, toxins, or other peptides. Evasion escape from the host cell, actin polymerization, destroy membrane significantly by the secreted proteins. So protein A is an anti-complementary since binding to IgG is a complement cascade is activated, depleting complement level. Thus, in the presence of a protein A, interaction of bacteria via bound complement with C3 receptors will be inhibited. Free protein A binds to the Fc portion of IgG, thus phagocytosis via Fc receptors may not occur because of steric hindrance. So, bacterioglycan, like lipopolysaccharides, can activate the alternate complement cascade. In S. Pyogenes, peptidoglycan is sufficiently exposed that it is able to bind to complement. The M protein of group A, streptococci, is antiphagocytic uh, component of fimbriae. M protein binds to fibrinogen from plasma, which blocks complement binding to the underlying peptidoglycan layer. Thus, streptococci in non-immune serum uh, are not phagocytosed intracellular pathogens, both including obligate and facultative, must be able to avoid being killed within the uh, phagosomes. This occurs from bypassing the lysing these vesicles and then residing free in the cytoplasm. Alternatively, they can survive in phagosomes. Fusion of phagosomes with lysosomes may be inhibited, or the organisms may be resistant to degradative enzymes if fusion with lysosomes occur. So, bacterial pathogenesis and antibiotic resistance are growing problem. Across the span of human history, pathogenic bacteria have been one of the main killers of human and animal population, causing untold misery to human civilization. Throughout the 20th century, medical science managed to make significant progress against pathogenic bacteria and the disease they cause through the use of antibiotics. However, due to antibiotics being overprescribed by medical professionals and the overuse of antibiotics the within the dairy meat industry and several other worrying setbacks in the fight against the bacterial infection. New drug-resistant strains of bacteria, including MRSA, have emerged over the past few decades, and old diseases such as tuberculosis have now adapted and are on the rise in developing countries, where previously they were more close to eradication. Currently, medical professionals are researching several alternatives to antibiotics to increase the effectiveness of medical science in combating infection. So, how does antibiotic resistance occurs? So, the first thing is that a high number of bacteria, a few of them are resistant to antibiotics. The second is antibiotic kills the bacteria causing illness as well as good bacteria protecting the body from infection. Third, the resistant bacteria is now have preferred conditions to grow and take over. Fourth, bacteria can even transfer their resistance to other bacteria, causing more problems. In conclusion, we can say that the relationship between host and pathogens is a complex one that is constantly evolving. Pathogenicity depends on a variety of factors, both belonging to microorganisms and the host, and any perturbations in this relationship can have profound effects on the progression and severity of the disease caused. The vessel modern medicine has managed to effectively treat bacterial infection, growing issues such as antibiotic resistance show that there are clearly some ways to go before we can completely eradicate the disease. So MCQs. Uh, which of the following would be a sign of infection? The correct answer is C. Fever. Which of the following is an example of a non-communicable disease? B. Food poisoning uh, due to a pre-found bacterial toxin in food. During an oral surgery, the surgeon nicked the patient's gum with a sharp instrument. This allowed Streptococcus, a bacterium normally found inside mouth, to gain access to the blood. As a result, the patient developed bacterial endocarditis, an infection of the heart. Which type of disease it is? Heterogenic. Which period is the stage of disease during which patient begins to present general signs and symptoms? Prodromal, a communicable disease that can easily be transmitted from person to person, is which type of disease? Contagious. Which of the following is a pathogen that can not be identified by the original cause postulates? HIV or human immunodeficiency virus. Uh, One of the advantage of combining vancomycin with uh, linezolid is less nephrotoxicity. The most common pathogenicity of pneumonia is a microaspiration of upper airway secretions through inapparent aspirations. The traditional approach of anti uh, drug combination is anti-pseudomonal beta-lactam with aminoglycoside. A major factor that increases the resistance of gram-negative bacteria is both A and B prior to broad-spectrum antibiotics and immunocompromised person. These are the references from where we have collected the material, and thank you very much for listening to my lecture.